Hello dear students, today is our day 27, video 16 and for class 12. Before we proceed towards megasporophyll, we must study the recap videos. Recap of our second chapter, that is sexual reproduction in flowering plants, in which we studied about microsporophyll. Fine. So first of all we studied about microsporophyll, and the microsporophyll is the stamen. Stamen has two parts, one is the anther and the other is the filament. Anther is bilobed and each lobe contains two theca, that's why the anther is known as diathecous. In that case, what we studied about microsporangium. In each theca, two microsporangium are present. As anther is diathecous, so they are having four microsporangium in four corners. And these microsporangium are storing a homogeneous mass of tissue called as sporogenous tissue, whose each and every cell is capable to behave like pollen mother cell for the formation of microspores. So these microsporangiums are called as pollen sacs also. After that, we studied about microsporogenesis. Microsporogenesis is what? In case of microsporogenesis, the process of formation of microspores from pollen mother cell through single meiotic division. One meiotic division give rise to 4 microspores. Fine, these microspores are actually the male gametophytes. These microspores on maturation, they are called as pollen grains. Fine. Then we studied about the structure of microspore. The microspore is surrounded by two layers, two wall layers. One is the exine, that is the discontinuous layer, intine, which is the continuous layer. The discontinuous layer is made up of sporopollenin, and intine, the continuous layer is made up of cellulose and pectin. Sporopollenin, because of the presence of sporopollenin, pollen grains can be stored for longer periods through a process called cryopreservation, in which we preserve pollen grains in liquid nitrogen at minus 196 degrees Celsius. After that, we are having another video in which we studied about pollen grains. The pollen grains on dehiscence. After the dehiscence of the anther, pollen grains are released into the external environment, and during their release, in case of 60% of angiosperms, they are released in two cell stage, and those two cell stage bears two types of cells in a single pollen grain. One is the generative and the other is the vegetative cell. And in case of 40% of angiosperms, they are shed in three cell stage, and the three cells stage contains one vegetative cell and two male gametes, right? So here, for the formation of mature pollen grain to form or to move into the female gametophyte, they perform two mitotic divisions. Fine. Next, we studied about additional features of pollen grains. These pollen grains, apart from acting as male gametophyte, they are having certain other functions too. They are causing allergy in few individuals. For example, we studied about Parthenium, which is causing allergy like asthma and bronchitis. These both are the respiratory disorders. Secondly, they can be act as a nutrient in the form of syrups or tablets to enhance the performances of racehorses and athletes. And third thing that we studied about pollen grains is their ability to germinate over a specific stigma, which is most few of the pollen grains can remain viable for months, or few lose their viability as soon as they are released, or some of them can retain their viability up to 30 minutes of their release. Fine. So this is a recapitulation video of microsporophyll, or the formation of male gametophyte, where for the formation of male gametophyte, it will undergo one meiotic division and two mitotic divisions. ठीक है, इस चीज को particularly आपको याद रखना है, क्योंकि this will comes, ये आपको हेल्प करेगा आगे न्यूमेरिकल्स को सॉल्व करने में. ठीक है, आज ही मैं एक और वीडियो आपके पास शेयर करूंगी, that will be regarding our megasporophyll. Fine, thank you.